proteins, enzymes, and we start replicating the DNA or the RNA if it's going to be an RNA virus as well. Then we reassemble those pieces, and then the virus is finally going to be released outside the cell. So one of the differences here is that it goes on to talk about phage, right? Bacteriophage, viruses that infect bacteria but not animal cells. Phage have a similar set of stages here, right? Only we don't see uncoding. Sorry, I was trying to think. Which one is it? We don't see the uncoding when you're talking about bacteriophage. So it's something to highlight. Make sure you know. The reason why we don't have uncoding with bacteriophage is because the capsid layer stays completely outside the cell, and it only injects the genome inside the cell. So we don't have an uncoding stage during bacteriophage replication. Okay, so let's look at synthesis events from the perspective of, I don't know, you guys can't see very well, but if I go over there, you're really not going to be able to see. So I don't know. I can't get this video down. Can you see it here at least some? From the perspective of the central dogma. We have DNA replication. We have, I'm skipping a few stages here. With functional protein, right? This is gene expression. Okay, so this is what's happening in your cell, right? DNA replication, we said, is under the control of the major enzyme is DNA polymerase. Remember last time we said DNA polymerase is a DNA-dependent DNA polymerase. It binds to template DNA to read and make a complementary molecule of DNA. Read DNA and produce DNA. The major step in transcription, one of the major steps in gene expression, transcription, we use an RNA polymerase. RNA polymerase is DNA dependent, binds to DNA, but produces an RNA molecule. So it is a DNA dependent RNA polymerase. And those are the two major enzymes that we have that are working with replication and gene expression. Because I mentioned, I think last time, that viruses sometimes have to bring in their own polymerases. They can't use the polymerases that we have. This happens to be all of the RNA viruses that fall in that category. The DNA viruses can use what we have. Okay? Messenger RNA to be produced into a protein. What organelle are we talking about here? In this step of translation. Ribosomes, right? Translation occurs on a ribosome. We want to keep that in mind as well. So, two things have to occur during synthesis for a virus to be reformed. One is we have to take its genome, its genetic material, and again, viruses can be either DNA or RNA. We have to make copies of it. We have to replicate it, so we have a lot of copies of that genome. And then viruses have to produce their viral proteins, some of which are capsid proteins that package that genome back up so it can be released from the cell. So we're talking about more genetic copies of the genome, and we're talking about viral proteins. Both of those have to be produced. Well, you know that those happen in different areas in our own cells, right? We don't replicate our DNA by binding to a ribosome. That's a completely different function. We produce proteins from our ribosomes, right? So the viruses are gonna take advantage of our enzymes and some of our organelles involved in this. And we'll start with the easiest one to talk about, which is a double-stranded DNA virus. And double-stranded DNA virus, I'm going to try to give some examples of what this might kind of look like. But let's say ATGCT, let's say that's the genome, but it's a double-stranded DNA virus, right? So we would have the complement TACGA, right? You all with me?
Mm-hmm.
How we formed a complement?
Mm-hmm.
Good. So the virus comes in, and obviously there's thousands of nucleotides in the viral genome. I'm just keeping it very short here so you can see an idea. It comes in, all right, and it has to produce proteins off of its genome, and it has to produce more of its genome. How is it going to do this? Well, these strands can be separated just as we separate our own DNA, and this organism will use our DNA polymerase, okay, to produce complements of those strands. So now we have this ATGCT producing a new complement, TACGA, and this TACGA producing its new complement, ATGCT, by using our DNA polymerase. And it's going to do this over and over and over and over. So 1 becomes 2, 2 becomes 4, 8, 16, 32, 64. We get a mass amount of viral genomes very quickly. That's one step, okay? The other step then is to take this information here in the genome and produce proteins from it, viral proteins that are essential in reassembly and release. Well, one of these sides is going to be known as the template side that has that information. And as we do in our own cells, that template side gets read in the 3 to 5 by an RNA polymerase, and it produces viral messenger RNA. That viral messenger RNA now binds to our ribosomes and produces viral proteins. So I want you to think of these synthesis events as two things going on. One, we have to produce more of the genome from what we're given. Two, we have to produce the viral proteins from what we're given. And if you're given a double-stranded piece of DNA, it's pretty straightforward. It's really close to what we've got going on in our own cells, since we're double-stranded DNA. And we just use, or the virus just uses all of the polymerase enzymes and ribosomes that we have to produce its genome and proteins. Does that make sense? Okay. So we're just going to keep playing on this theme because viruses like to take advantage of us, our stuff, what we have going on in our cell. The next one is a single-stranded DNA virus. All right, some viruses out there just have a single strand. I'm not even going to erase this. All I'm going to do, or try to do, is back up and put ATGCT up there and put single-stranded DNA virus. This is what we're starting with. The first step that has to occur is to produce the complement to this. And when you produce the complement, you now form the double-stranded DNA molecule. Okay, what if I want to form the other side, which is this, to my single-stranded DNA virus, what enzyme am I going to use? What can make DNA from DNA?
DNA polymerase.
DNA polymerase. So the host DNA polymerase will make the complementary side so we have a double-stranded DNA molecule. Now, we can start making both sides again if we need to. We don't really have to since we just need to keep a single strand. But we've got the other side now to produce more of the single-stranded DNA. Then this molecule up here can be read by our RNA polymerases to produce more of that message and to produce more of the viral proteins. So being a single-stranded DNA molecule, it's just one more step really. You gotta produce the complement
By using DNA polymerase.
by using
Okay.
that same DNA polymerase, yep. So none of those DNA viruses have to bring in their own polymerases or enzymes. They use all the host enzymes, the host ribosomes, to produce what they need. Unlike these guys here, all of the RNA viruses have to bring in their own RNA polymerase. Their RNA polymerase is RNA-dependent RNA polymerase, meaning it reads RNA and produces an RNA molecule. Some of these RNA viruses, the single-stranded ones, are considered positive sense single strand. Some of them are considered negative sense single strand. Okay. So positive sense RNA virus. Where do I have it at? Well, I did that wrong. I put T's in there. Can't do that with an RNA virus, can you? No thymines in RNA, there's only uracils. So AUGCU is our positive single-strand RNA virus. If I'm going to make more of this genome, what do I have to do? Can I just read AUGCU and
Do
have a
the complement?
polymerase that makes AUGCU? I have to make a complement first. So there's kind of an intermediate step. I can't do a straight leap of faith here. I have to make the complement. So this single-stranded RNA virus produces UACGA. This gets confusing after a while. Right? This is now what's known as the negative form of it. The complement to it. Okay? And now, from this negative sense, we can start producing numerous positive sense genomes. Okay? So, positive RNA virus, we have to read the positive sense and make a negative. Now we have a template to make all the positive sense genomes that we can possibly make. All right? Problem. How do you use one of our enzymes, read an RNA message, and produce an RNA molecule?
RNA.
You don't.
Yeah.
We don't have anything that can help them.
You don't.
Again, all RNA viruses have to bring in their own RNA polymerase to do this. So this is an RNA-dependent RNA polymerase that we don't provide for them. So they can take this RNA molecule and make a complement RNA molecule. And then they can read this RNA molecule and make the positive RNA molecules. All right, so our genome is done. Now we got to produce the proteins. Like I said, we've got genome to worry about and viral proteins to worry about. Positive sense, what this truly tells us, the importance of this is that when this is a positive sense molecule, it can be directly interpreted by the ribosome as is, and functional proteins start getting produced. This looks like our own message, okay? So immediately the positive can be read by our ribosomes, and viral functional proteins start getting produced down here. All right? If you are a negative sense RNA virus, we have to back it up. take off these because these are kind of in the way. Make confusion there. All right, here we are. Negative single-stranded RNA virus comes in at UACGA. What this tells us, because it's negative, is that if this bound to a ribosome, it would not be read. Only positive sense will be read by ribosomes directly. So, how do we get those proteins to form? We're going to make a positive sense out of it first. So again, this guy brings in its own RNA polymerase to take RNA and make a positive form of RNA. All right? And now this can be read directly by our ribosomes, produce viral proteins. This can also be used as the template to produce more negative single-stranded RNA viruses.
How do you know if the original is negative or positive? Maybe I'm not understanding.
You don't have to.
Okay,
It's the type of virus that's deemed a negative versus a positive.
okay.
When they explore their replication cycle, if they see this occurring before that, and there's ways scientifically to do this, they know it's a negative form of it. They also can look at the genes and say this would not form any functional type of protein, but if we did the complement, we know it would. There's
so
different
it
techniques
has
where
to
they
do
know it.
with the actual, like,
The sequence,
codons,
Yeah.
sequence
it has to
of
do with
the,
sequence.
okay,
Yeah, this doesn't make sense to form a protein.
okay.
This form of it would. So if it comes in looking like this, the virus knows what it has to do. It's going to form positive RNA, and that's going to be read by the ribosomes to produce the proteins. Now that positive also acts like a template to make more negative genome. Okay? The last one type of virus, well, there's another one beyond that, but the last RNA virus that kind of follows this pattern is double-stranded RNA. And this one, I think, is pretty more straightforward. You basically have both sides of it to begin with, right? So regardless if that's your negative side, you would already have the positive side to it as well, since it's a double-stranded RNA virus. Okay? This would be used to make more double-stranded RNA as well. This side of it can be used to be read by the ribosomes to produce more of the viral proteins.
No matter if it's the positive RNA, the single-stranded, negative, or the double-stranded, will that step occur at the same time where the positive will?
Yeah, don't get tied up with the
That step,
sequence
okay.
of events. No, think more about how does this work.
Okay.
Because honestly, sequence of events would probably be none of this really occurs until we produced a bunch of genome pieces.
Okay.
Then once we have a certain level of genomes, some of those will be used to
Okay,
produce
so
the
don't worry
proteins.
about that
Don't
part
worry about it, yeah.
itself?
Don't get
Okay.
tied up with that. That's not important. We can see it. Yeah, because if you had a single strand of this come in, like a single
Yeah.
molecule of this would never occur until we got a bunch of more of
And
this.
enough of them?
Yeah.
Okay.
But that's not really important. I just want you to see how we're doing it, how the virus does it. So again, RNA to RNA still need its own polymerase, right? So all the RNA viruses bring in their own RNA polymerase for copying their genome. This is a good illustration. Walk yourself through it. Think about what's going on. It breaks it apart into two sides. One is to produce the genome, and one is to produce the viral proteins. All right, and let's see what's going on as far as illustration is concerned. The last virus to talk about and are called, the group's called retroviruses. Now HIV falls into that group, but there are other retroviruses other than just HIV. But the term retro means to go back. Doesn't necessarily mean old or vintage. It just means to go backwards, okay? And these guys come in as single-stranded RNA viruses Yeah, it's a positive RNA viruses of HIV. But anyway, instead of making more RNA, it tends to go backwards and make double-stranded DNA from its single-stranded RNA molecule. To do this requires an even more special enzyme, which are simply known as, the classes of them are known as reverse transcriptase. It's a type of polymerase. It goes in reverse. It makes a transcript, but it makes a DNA transcript from that RNA molecule. So we have a reverse transcriptase enzyme, or RT, right there in those retroviruses. Okay. Then with HIV and many of the retroviruses, the double-stranded DNA virus will actually incorporate into the host DNA and become part of your chromosome. Right? It inserts itself into your chromosomes and can produce the viral proteins by that mechanism. At some point, it can induce and create more of the RNA genomes as well. But it overall is using your stuff from that point on. Is the point here. It's using your polymerases, your ribosomes to produce what it needs to make more of those viral particles. So it really just requires its own reverse transcriptase to get it started. And that's what we've focused on with HIV treatments. Reverse transcriptase inhibitors, in fact, is one of the variety of types of drugs that are used to keep HIV in check. If we can block this step, even though it's infected in your cell, even though the genome is in your cell, it does not do this. It's not how it works. It's not going to work that way. So if we can block this step, it never becomes double-stranded DNA, and so it won't produce any more of the HIV particles. Okay. So retroviruses just kind of go backwards. They require a unique polymerase for that step to occur.
So morally, if RT never occurs to produce the double-stranded DNA, it won't produce the proteins? It
Yep.
never occurs? Okay.
Nope. You're going to have very few molecules of this. Even if this does produce some of the viral proteins, you're not increasing the overall load of genome. You're not increasing the number of viral particles.
It won't be enough.
Yeah, you're not producing genome. This will never go through another route to produce more of that RNA. It has to go through that DNA route first. Questions? Okay. That's kind of the only things I was going to focus on today. The synthesis events. I'm trying to think. See, here's the bacteriophage, or what's known as phage. These are viruses that only infect bacteria. I don't know if this is on there or if I took it off, but I talk about it. But there is something known as phage therapy that's slowly coming about. I don't know if you've ever heard of this. But I used to spend quite a bit of time in class discussing it with students. But since we have viruses that just infect bacteria and not your own cells, right? They can only infect bacteria. Someone thought, hey, wouldn't this be a unique type of therapy since our antibiotics don't work all that well anymore? So they've slowly gotten around to start using phage as a mechanism to kill specific types of bacteria. They're very bacterial specific, which is nice. So like unlike antibiotics, you take a drug and all your bacteria in your body is wiped out. This only targets the sole source of that infection there, these types of organisms. In the States, this has actually been around, phage has been around, and the therapy has been around since the early 1900s. But then someone came across antibiotics, and everybody went, woo, we got antibiotics, and World War II started, and we needed drugs right away, and phage just kind of got tossed to the side. But other countries like Russia, countries in Africa, have been using phage therapy this whole time and don't depend on antibiotics whatsoever at all. Now, because we've pharmaceutically developed so many antibiotics, we are not getting approval fast enough for phage, even though we know it works well because there's all these other patents for antibiotics that they haven't run out. But unbeknownst to me, a couple of years ago, and I'll stop rambling in a second, I came across an article and had no idea that we did do this in our country. They allow phage to be sprinkled on, or I say sprinkled lightly, not like a light dusting. I mean, like, I don't think they actually just sprinkle the phage on there. It's not like a salt shaker. On ready-to-eat meats, RTEs is what they call them. Hot dogs, you don't have to cook a hot dog. I never did as a kid. You just pull it out of the package and start eating it. Those are what you call your ready-to-eat meats, lunch meats, stuff like that. Well, one of the biggest issues if you're pregnant is what? With ready, I mean, no, there's a lot of issues, but with foods in general, refrigerator foods, ready-to-eat meats, listeriosis.
Mm-hmm.
Yeah. So that was one of the big organisms that they're having issues with on these ready-to-eat meats. You and me, healthy, maybe you feel sick for a day or two. Developing fetus, serious damage. Okay? So they found a phage that takes out listeria, and they sprinkle it on our ready-to-eat meats. And I never knew this, had no idea. And they've been doing it since, I don't know, probably the 90s. So we do have, while we don't supply treatments to people, we've been using it in consumer products. We've been using phage to combat bacterial infections before they ever
Does
get
that mean that the whole practice of not eating lunch meat when you're pregnant is kind of not really relevant anymore?
No, I wouldn't say that at all. Because even though it's on there and it's going to reduce, it may not completely control it. And so there's still a risk factor associated with it. I don't know, I haven't looked, but I have a feeling if you look at the ingredients on a package of hot dogs, it doesn't say phage, bacteriophage, right?
Probably not.
Slightly sprinkled.
Light sprinkle.
Lightly sprinkled.
Is it probably different in the Great Value brand? than-
Yeah.
Oh, yeah.
Of course.
Much cheaper version of
Less phage,
$1
not a great
off.
value brand.
It's only 99% effective, not 99.9% effective.
Darn. All that.
Yeah,
$1.
I thought that was crazy.
Yeah, that I didn't know that.
Yeah, that's weird. So what about like meat that you get sliced at the deli?
I think that would still be considered.
Would that be considered ready to eat, even though they just slice it in
Yeah,
front of you?
I'm trying to think.
I think
mean,
it's
not if you're like
less.
doing prime rib or something, but if
Yeah.
you're talking
Oh, yeah,
about lunch
no,
meats,
like-
all your lunch meats are already
Ugh.
cooked and ready to be eaten.
Never eating a sandwich again.
But the whole slicing thing bothers me to begin with. I don't clean those things very well.
No. Mm-mm.
So, but wouldn't it be nice to have your own little container of phage?
Sprinkle it on everywhere?
On everything?
You think they'll use this to replace antibiotics at some point?
I think, well, they've already started in our country now. They're doing it with certain very specific things. Like the thing I read was about like an aortic repair or something. I can't remember. It might have been
Just
a heart
open you
repair.
up and-
No, I can't. And there's a certain type of infection that's highly associated with that specific maneuver or whatever. So they've approved a specific phage to be taken at the time. And you take it like anything else. Sometimes it's up the nose, sometimes it's a liquid that you drink from a vial, and it does no harm to you. It only targets the specific bacteria, and that's it. And it's cheaper than antibiotics too.
Yeah.
Oh, we
They
just
made
take way
faster.
too many antibiotics now, so they just don't work.
Yeah.
And
Or
even
you get
though
used to
you can, they have shown some phage resistance, they can quickly genetically modify the phage, whereas we can't quickly create a new antibiotic. So we can combat that.
So this would be, you would, I mean, eventually, are people gonna just become so antibiotic resistant that this is gonna be the only way to really target stuff anyway? I mean, I feel like antibiotic resistance.
If they take them away, they're taken.
Yeah, the evolution of it is interesting. Once we all become so resistant to it that they stop prescribing the antibiotic, then after a while, we'll see a decrease in resistance to that antibiotic again. We
Mhm.
just have to starve them of that, and then they'll lose the resistance gene over time, and then we can start using the antibiotic again. But there really is something to say that, like, let's not just sit there and take antibiotics for every little cough, sneeze, cold, because it doesn't target what you think it's targeting anyway. It's doing more harm to your body by taking those antibiotics than, you really should only take them when you need to.
Right.
So, anyways, I need a break.
So,
Cause I got another class.
this is like an off-topic question, but it's just curiosity. So, say you have, like, knee pain, and people are like,